Okay, thank you everyone for joining meeting number 12 of the M2D2 series. We are very honored to have Octavian here with us today. About Octavian, Octavian is a postdoctoral researcher at C-Cells MIT, working with Tommy Giacola and Regina Basile on deep learning solutions for drug discovery and structural biology using geometric and physical induced biases. He received his PhD from UTH Zurich under the supervision of Thomas Hoffmann, working on non-Euclidean representation, learning for graphs, hierarchical data, and natural language processing. He is also a member of machine learning for pharmaceutical discovery and synthesis consortium, the Abdul Latif Jamil Clinic for Machine Learning in Health, the DARPA Accelerated Molecular Discovery Program, and the ELIS Society. His research includes Spotlight at ICLR 2022, Spotlight at Norris 2021, 2028, and oral talks at ACML 2018 and 2019. Without further ado, let's welcome the speaker. Thank you again, Octavian, for being here today, and the floor is yours.
Thank you, Prudencio, and thank you everyone for taking the time to attend my talk. So I want to make it like an interesting discussion, so feel free to jump in with questions. I have quite a little bit of content, so if you wish, we can delay the questions for the very end, but otherwise, let's dive into it. So I'm going to talk about how we can combine deep learning and geometry to model 3D structures and 3D interactions of molecules. So I want to acknowledge my collaborators, which are listed here before I start. So why are we interested in 3D structures and interactions of molecules? So this problem is a key aspect of understanding how diseases act, like the mechanisms of diseases in our body or animal bodies, and also to accelerate drug discovery. So I want to show here just a motivating video where we see abnormal protein-protein interactions. So we have these proteins which are on the surface of tumor cells, and they interact, they form complexes. And this actually triggers then various other effects, such as tumor growth or metastasis proliferation. So these abnormal protein-protein interactions happen for diseases, but also protein-protein interactions happen in all over the human cell. So it is very important to understand those also from a 3D perspective, understanding how proteins deform and where in how they actually attach to each other. So there has been this therapeutics idea, which is tried a lot in pharmaceutical companies. Essentially, we know proteins, how they interact and in which parts they interact, sometimes for some proteins, and we want to prevent their formation. So maybe we can design new molecules, such as an antibody shown here, to prevent the formation of such complexes. So in this talk, I want to present my recent models, which accelerate drug discovery. So the goal here is to design fast and accurate AI models that are going to predict complexes of molecules and also 3D structures of molecules. So I'm going to focus on this problem of we have as input two molecules, maybe two proteins, or maybe a protein and a drug-like molecule, and we want to predict how they attach to each other. So we want to predict the 3D structure of the complex. And I'll also highlight a solution for how we can predict conformations, like ensembles of low energetic conformations of small drug-like molecules. So I combine geometry and deep learning to tackle these two problems, and I achieve like between tens and hundreds of times speed ups comparable when you compare with existing solutions like commercial and open source software, while achieving comparable or better quality, as I will show a bit later. So previous methods are insufficient. I will show that they are slow and inaccurate because they have to explore a very vast space, and they do that in a rather inefficient way. So you might ask, why do we need speed? And so speed is actually key for various aspects of drug discovery pipeline. So one is virtual screening. Let's say we have a target, which is a protein, maybe a cancer protein, and we want to find the best set of drugs or drug candidates that are going to attach to the specific target and maybe inhibit its activity. So for instance, we have data sets of billions of millions or billions of synthesizable drug-like molecules, and we want to scan them very rapidly and find the possible candidate molecules. So we need to have very fast computational methods in order to do that. We also want to, like, once we find good drug candidates, we want to scan computationally for toxicity. So one aspect of toxicity is to discover side effects of therapies in the sense of like off targets. So a drug can attach to a target, which is, let's say, a cancer protein, but it can also attach to an off target, which is a protein from a normal tissue that we will not want it to attach to. So one way to do that is to scan the entire human proteome, or at least the subset for which we know the 3D structures of the proteins, and then see if our drug candidate can potentially also attach to some off targets, and then maybe go ahead and in wet lab experiments test this hypothesis out. So I'm going to show some representations of proteins and molecules, and just to make sure that everybody in the audience is on the same page. So I just want to highlight the fact that drug-like molecules can be seen as graphs, so molecular graphs where nodes are atoms and bonds are edges. But in fact, in reality, they are 3D point clouds, 3D objects. We can view them as 3D objects, which have actually flexibility, internal flexibility in, let's say, torsion angles of rotatable bonds. So for one molecular graph, in fact, in practice, we have an ensemble of many low energetic states. And these are two possible views of drug-like molecules. When we talk about proteins, we have proteins that represent the sequences of amino acids or residues, which are small molecules. And these sequences are folded in 3D. From a computer science perspective, we can view them as 3D spatial graphs. And this is the view we are going to adapt today. And we can also view them as meshes of irregular surfaces of some 3D object. So you can imagine we can tackle these problems using various techniques from maybe NLP or graph representation learning, and this is where my core domain area is, and also geometry processing, computer vision, 3D graphics, and so on. So when we talk about 3D structures and interactions of molecules, we know that they are governed by laws of geometry, physics, biology, and chemistry. So unfortunately, previous deep learning models that deal with these problems, they struggle to learn these principles implicitly, because you will see a bit later, we have insufficient data compared to other deep learning data modalities such as text or vision. So here we talk usually about a few thousand of training samples. So this is very little amount of data. And the goal here is to incorporate as much as possible from prior knowledge in principled ways into these deep learning models, and I call those inductive biases. So these are priors, for really domain knowledge priors, and I'll give some examples just in a second. And the goal to incorporate these priors into our deep learning solutions is to build trust. So if we now take our models to industry, to pharmaceutical companies, we want to show why we want to show that we incorporate some of the domain knowledge that is known for decades. Okay, so some examples of such inductive biases are shown here, but of course it's not exhaustive. It's just a set of small examples. So symmetries, for instance, we know molecules are invariant with respect to rotations and translations in the Euclidean space, which means that if we rotate or translate a protein, let's say, we are going to change the numerical representation, but we are not going to change the underlying biological object. This is, for instance, not true for reflections, which are going to change, so are going to affect aspects such as chirality. And I will tackle that a little bit later. So we also talk about independent symmetries in multi-body interactions. And in today's talk, I'm going to touch on this aspect a bit more in detail. We have also geometric constraints. So for instance, when we talk about proteins creating a complex, we know that protein volumes cannot intersect. And in fact, they should be actually tangent during binding. So we want to have this constraint. We also have chemical constraints. So we know certain residues are more hydrophobic and others are more hydrophilic. And the hydrophobic ones, they want to minimize their contact with water molecules. So water molecules surround proteins in the human cell. So what happens is that these hydrophobic interactions, they tend to cluster. And in this way, they minimize their surface contact with water molecules. And this aspect is important both for, let's say, folding, protein folding, or molecular, also for protein or molecular interactions. So we also have physical constraints, for instance, various forces, intermolecular forces such as van der Waals. So in my research, I create deep learning models by leveraging powerful tools from various other domains. For instance, optimal transport, which was developed as a branch of mathematics, and then recently adapted quite a bit in machine learning and in various aspects of machine learning. Of course, equivariance to Euclidean symmetries, and we'll talk about that in a second. Graph neural networks is one of the core technologies I use to embed graphs. And then various aspects of geometry, 3D geometry, such as geometry of tangent and non-intersecting bodies. So with that, let's dive into the first part of this talk, which is called EquiDock. So the idea here is we have two proteins as input, and we know that they are going to interact. And what we want to do is to predict the final complex. So essentially, what we want to do is to take these two structures of the two proteins as input and have a neural network, which we called equivariant docking, so EquiDock. And it's going to predict, for now, it's going to be a rigid transformation of one of the proteins, which we call ligand, to place it in the right location and with the right orientation with respect to the second protein. And in this way, you predict the final protein complex. So in this problem, we call it blind docking, because we don't take any assumption of the areas of where the two proteins are likely to interact. So the binding site. So we have several challenges of previous docking methods. So they are typically very time consuming, and we talk about tens of minutes to hours per single complex. And why that happens is because they are actually facing an enormous search space of possible ways to attach the two proteins. So how these models typically work is that they take the two proteins and place them in essentially all possible ways to attach them. So they sample like millions of such candidates, maybe using some Monte Carlo search. So they have this expensive candidate sampling. And once they have, this is followed by a scoring procedure, which can involve deep learning or can involve more traditional methods such as force fields. And then it's followed by ranking. And finally, the top scored candidates are then further fine-tuned in their respective binding locations. So instead, my deep learning model is going to do that as a direct prediction of the protein complex in just seconds. And I'm going to show how that happens. But the idea is that we are going to bypass this expensive candidate sampling strategy. So a key aspect is one geometrical constraint, which is this invariance to initial placements, orientations, and roles of the two proteins. So if we have, let's say, the ligand initialized in 3D, like placed randomly in different locations, maybe rotated differently, we want to make sure that the final protein complex is predicted always identical, no matter where the ligand is placed in space, even if it's placed like to overlap with the receptor, or if it's placed like 100 angstrom apart. We also want to be invariant to this notion of roles. So which one is a receptor or which one is a ligand should not matter for proteins. It would matter for drug-like molecules. And that I'm going to discuss a bit later. So in order to do that, like previous methods have relied on strategies such as data augmentation. Unfortunately, this suffers from two issues. One issue is that it's actually very expensive because you have to increase the amount of training data by many times, and it also doesn't generalize to any new, any unseen initial positions, let's say at test time. So in our paper, we derive a theoretical result which gives us necessary and sufficient constraints that should be incorporated in all deep learning models that predict protein complexes. And we also show how to hard code those into deep learning models. So these are not just soft constraints like regularizer, but really hard constraints. And I'm going to explain how that works in our model. So, okay, so now let's dive into the core of this EquiDock model. So I'm going to first show the intuition and then get into the details. So at training time, we have access to these true docked complexes. So they have 3D structures of both proteins, and they are already placed in their co-crystal positions. So we have receptor and ligand coordinates, which in my case are going to be just coordinates of alpha carbons of each residue, but various other ways to represent 3D coordinates are possible. For instance, taking all atoms of proteins. So once we have such a complex structure, we are going to define the set of so-called binding interface points. So these are a set of points that lie at the contact between the two proteins. So this can be defined in multiple ways, but the way we define it in our work is to take pairs of residues from both proteins that are closer than a threshold. In our case, it's 8 angstrom. So we take all these pairs of residues. If you want, you can use it as a bipartite graph, and then we take midpoints of their segments in 3D, and these are going to give us a set of these red points, which we call binding interface points. And I call them P1 star equals P2 star because I view them as two identical sets, which are overlapping completely. But now, so this is like the same figure. So I call them P1 star equals P2 star, but now once we separate these two proteins out, we're going to have two sets, right? We're going to have P1 and P2, and these are going to be the undocked or unbound input structures. And what is very important here, and that's the main intuition of our model, is that if we now superimpose P1 and P2, we are going to, so essentially takes P1 and P2 and finds the rotation and translation to place them back together. Then we're going to recover the docking transformation to dock the two proteins back. So if we take R and T, which are the rotation and translation for superimposing P1 and P2, and we apply it now to this ligand protein, we're going to recover the true complex. So, but obviously, we don't have P1 and P2 at test time, at inference time, right? So we only have it because of the training data. So what can we do? So the idea here is that we want to predict some proxy set of 3D points. We are going to call key point sets. So they are denoted by Y1 and Y2. And we want Y1 and Y2 to approximate well the binding interface points, P1 and P2. So if we have that, we are going to now then, instead of superimposing P1 and P2, which we don't have at test time, we're going to superimpose Y1 and Y2, and we're going to recover the docking. We're going to predict in this way the docking, rotation, and translation to dock the two proteins. Okay, so the idea is to use a deep learning model to predict these 3D key point sets, Y1 and Y2. So I'm going to show first what is the architecture, the deep learning architecture to predict Y1 and Y2, and what kind of constraints we want to impose there. And then I'm going to show what it means exactly this approximate P1 and P2 sentence here. Because that's currently a little bit ambiguous because we have to deal with sets of unordered list of objects of 3D points. Okay, so the architecture here is the following. So we have two proteins as input, and we want to have a deep learning model. We call it F, which is going to predict key point sets. So in this case, it's going to predict, let's say, just in this cartoon setup, it's going to predict 3 black stars and 3 blue stars corresponding to each of the two proteins. So here is where the first aspect of symmetries and equivariance has to be incorporated in the model. So when we talk about group equivariance or Euclidean symmetries incorporated in deep learning models, we have in mind this classic, already like becoming very classic model of convnets, which takes as input an image and applies a deep learning model to do maybe image segmentation. And now if we apply an Euclidean transformation, so we slightly move, we translate the object in the image, and then we apply the same deep learning model. What we want is to guarantee that the output of this deep learning model is such that if we invert the initial Euclidean transformation, we are going to recover the output of the first image. So essentially, this was incorporated in convnets, as I said, and in various other architectures. And this has been also generalized to 3D point clouds, so 3D objects, and also incorporating not just translations, but also rotations and maybe reflections. And there is also, of course, the more theoretical notion of group equivariance. So we can generalize this to not just Euclidean symmetries, but any type of group actions. So what you can see here is that this is only acting on a single object. So in our case, we have to deal with multi-objects. So we have two objects as input, and this can be easily generalized to multiple objects. And we talk about this notion of independence equivariance, and I'm going to explain that in more detail in a second, but just the intuition of it is shown on this slide. So if we take these two proteins, we have some key points as output. And now if we randomly rotate and translate the two proteins, we want to guarantee that the key points are going to exactly and deterministically follow how these two proteins were initially rotated and translated. So this is pretty much the figure I showed before with a cat and the translation and a rotation, but now it's actually two independent transformations that change the two proteins. Okay, so that's the notion of multi-body equivariance. So let's see how we actually do that in our model. So we have this EquiDock deep learning model, which is going to predict two key point sets of equal cardinality as output. So the two input proteins are obviously going to have different number of residues or different number of points, but we always guarantee that the output is going to have a fixed number of points, let's say k in this case. So the idea here is to stack specially designed layers that are going to integrate Euclidean symmetries. And let's see how we actually do that. So we take the two proteins as input, and we represent them as spatial graphs. So simply putting it, we have this idea of k nearest neighbor individual protein graphs, which means that we take each alpha carbon of each residue to represent one residue. So we're going to retain the 3D coordinates of residues in this way. And for each residue, you also have various types of features, such as residue type. And then we have some local frame features also relating to the orientation of local frames of residues. And we also have pairwise edge features, which are actually not shown on this slide, but each edge also has some features. So we built in this way, we built a ligand graph and the receptor graph. So we connect each node with a 10 closest nearest neighbors. But again, yeah, this is like a hyperparameter. So so these k nearest neighbor graphs can look also differently depending on how one decides to create them. Okay, so here are the spatial graphs shown in a cartoonish way, just to make it a bit more simple to digest this figure. So we have these two graphs. Again, we have coordinates denoted by x, and we are going to have features which are going to be denoted by f or h. And then what we do is that we run message passing neural network, but we have to use a special type of message passing neural network. So usually MPNNs are acting on individual graphs. In our case, we have both intra messages like standard graph neural networks or standard message passing neural networks. But in addition, we also have cross inter messages, so messages between the two proteins. So if people are familiar with the work of graph matching networks, this is actually taking that one step further and also integrating symmetries and equivariance. So we have this idea of, so we call our model independent SHC equivariant graph matching networks. And let me say what this formally means. So what we have to do is to stack here several layers of this graph, of these message passing neural networks, and then transform coordinates and features jointly. And in this way, we are going to guarantee this core property of independent SSV equivariance of coordinates and invariance of feature embeddings. So this formally means that if we rotate and translate the 3D point clouds of the two proteins with completely independent rotations, Q1 and Q2, and independent translations, G1 and G2, we are going to see that deterministically in the output. So the output is going to always be transformed as such. But you see, like, this is very different from single objects. In single objects, you only have a single transformation of the original, a single rotation and translation of the input is going to affect a single output. Here we have multi-body. So let me just, I see there are a bunch of questions, so I will maybe, Prudencio, do you want to?
No, so basically I was going to say the same thing. There's a few questions that you might need to pay attention to. I can go through them quickly. So Simon Mattis was asking, was there any particular reasoning beyond going to KNN graphs versus thresholding the protein contact maps?
I'm not sure what you mean by thresholding the protein contact map. So in this case, we have the two proteins as input, and we don't know, it's a blind docking problem, right? So we don't know where they are going to interact. But maybe Simon, maybe you want to comment?
Yes, can you hear me?
Yes.
Ah, lovely. Yeah, I was just wondering. So you have the structures of the individual of the ligand and the receptor individually, right? In your problem.
Yes. Yes.
Yeah. So I was thinking you could use the contact map of this one, so the all-to-all distances essentially, and just say any residues that are lower than or closer than, say, 10 angstrom. We draw an edge between those. I was just wondering, was there any kind of consideration of graph connectivity?
So are you talking about cross connections between the two proteins?
Yes. So the way you built up your initial graph, that was, I was wondering whether there was any kind of special consideration why K and N.
Yeah. So in our case, so our architecture, as I said in the previous slides, is invariant to where you place the two proteins in space. All right. So in this case, there is no notion of what is going to be their proximity of the two proteins, because this is not, it's actually invariant to how the two proteins are initialized in space. I'm not sure if that.
Sorry, maybe I didn't express myself clearly. I mean,
yeah,
between residues in the same protein.
Oh, in the same protein. So okay. So what you're asking is if the k-nearest neighbor graph was not constructed in a way that. Sorry, can you say again?
Yeah. So what I meant was, when you built up the graphs to then do your message passing on them, it's basically as a pre-processing step, you have to define essentially where will you draw your edges and where not. And you at the moment, you say basically, if they are among the closest k neighbors, then I'll draw an edge. And so you know the distance between the individual residues to select the k closest ones. So I was wondering, equally as another prescription, you could also just say, if it's closer than, say, in a certain radius around this residue, I will draw an edge. And I was wondering,
sure.
if you looked at that.
So yeah, in fact, in our case, we have also a threshold for like 30 angstrom. So if it's what we do is that we take 10 closest neighbors, but in a radius of 30 angstrom. But of course, other possibilities are definitely here. Yeah. So we haven't played a lot with that aspect, but yeah, definitely your suggestion could be one good way to construct ligand graphs in a different way.
Awesome, thank you.
Sure.
Thank you.
So I see Dominique has a question. Do you encode distances as edge features? Yeah, we also have edge features which incorporate distances and also incorporate relative orientation of the local frames of the two residues, right? So each residue you can build a local frame, essentially like a local Cartesian coordinate system if you want, similar to what AlphaFold is doing, for instance. And then you can take the relative orientation of two local frames connected by an edge, let's say in the graph. And then you can build invariant features, SSV invariant features. So we actually do that. So it's actually all detailed in the appendix of our EquiDock paper. Andrea has a question. What exactly goes as an input into the point cloud is the protein coordinates only, or do you also use mesh information? Yeah, we also use some mesh information. We have, for instance, a very efficient feature that we designed ourselves to essentially as kind of a proxy for how close we are to the surface of the protein. So it's like a residue depth, if you want, in MSMS, for instance, software, if you're familiar with that. So again, yeah, this is all, and we have a few other features. Yeah, can you recall how the bipartite edges, or Vincent, sorry, can you recall how the bipartite edges or edges between nodes belonging to different graphs are constructed? Is it all pairs? Yeah, so for message passing, it's all pairs, but we use an attention way to send messages. So it's like graph attention networks, but for interconnections, right? Okay, so I see that there is something else. Okay, good. Someone else, or should I go?
Please continue. I think there's no more question.
Okay, awesome. So yeah, so here, yeah, so I already explained this idea of this core property of independent SST equivariance. So now the final step is to now take these coordinates, transform coordinates and embeddings, which are transformed jointly for the two proteins, and do some sort of projection to project them into two sets of ligand and receptor key points. And this is going to be handled by a particular instance of multi-head attention, which is going to incorporate SST equivariance. So just very quickly, this is using transform coordinates as values, and then keys and queries are just based on transform feature embeddings. Okay, so far I just explained the architecture of predicting key point sets, but what I didn't explain so far is now what it means actually that the key points approximate well the binding interface points, which again we have to do during training. So during training we have to train Y1 and Y2 to fit well into P1 and P2. But what does it even mean? Because we are dealing with sets of 3D points, right? So intuitively we have some constraints. I'm going to say why this problem is actually non-trivial. But intuitively, what we want is we want the distance between set Y2 and set P2 to be small. And also the distance between set Y1 and set P1, we want it to be small. Okay, so this is going to depend on some unknown alignments that we have to predict. So we have to predict which point of Y2 corresponds to which point of P2, for instance. And the same for Y1 and P1, right? So we have to predict these green dashes if you want. So in fact, these sets are actually not, so set Y, the key point sets and the binding interface sets are going to have different cardinality, right? So these alignments are not going to be bijective mappings, but they're going to be more like a sparse. If you're familiar with optimal transport, they're going to be sparse transport plans. And let me get back to that in a second. So as I said, we have to predict these alignments between key points and binding interface points in order to train our model. So this actually depends. So the first challenge here is that we have to be aware of this constraint, which is we know the alignment between P1 and P2. So we know, okay, these 2 points were originally identical in the docked position. We also know, so we essentially know this bijection between P1 and P2. We also know by design, because we use this multi-head attention layer, by model design, we know the alignment between key points in our models. We know the alignments between Y1 and Y2. So we have this constraint. The first constraint is that we want to respect the alignments Y1 and Y2 and P1 and P2. Now we have 2 more constraints. So, oh no, sorry. So what this means essentially is that if we're going to predict this alignment between this black star and this red dot, this is going to automatically imply the alignment between this blue star and this red dot. So we have to be aware of this constraint and incorporate it in our model. So a second challenge is that we want to make sure that the key points are going to cover well the binding interface points. So in this case, we cannot just align all black key points to a single red binding interface point, because in this case, this is not going to cover all the other remaining red points. So we really want to make sure that we do the idea of like essentially this set of Y2 is going to cover as much as possible all the points in P2, and is not going to left some over. And this alignment should also be based on 3D proximity. So ideally, we want to assign this black star to one of the closest red dots and not to the far one. Okay, so we have these three constraints, and the question is, how do we predict alignments in order to satisfy these three constraints, and also how we do it in a way that can be trained with our deep learning model. So our solution here is to predict jointly the two alignments, Y2P2 and Y1P1, via the idea of optimal transport. So optimal transport is a very nice way to mathematical formulation of matching two sets of objects, maybe sets of different cardinalities, and also in a way that gives us the matching, the alignment between elements in the set. So it both finds a set distance and as well as an alignment between an optimal alignment between points in a set, and it does it in a way that makes sure that one set covers well the other set. So it's this idea of mass. So if you have let's say some mass placed in all these black points, the mass is going to be equally distributed to all points in P2. So essentially, you can view this. So from a mathematical perspective, this T matrix is going to be a generalization of doubly stochastic matrices. So it's a matrix with positive, with non-negative elements, entries that is going to have identical marginals for rows and identical marginals for columns. So the way we do it, and I'm going to take a question in just a second, but I just want to say that the way we do it is that we have this cost matrix. So this cost matrix measures, is shown here, it measures the distance, so-called distance between every key point pair and every binding interface point pair. So the way it's done is essentially taking the 3D distance between a key point in the first protein and a binding interface point in the first protein, and the same for the second protein, and it's going to add them up. So what this T matrix is going to do is going to find a sparse mapping between indices of key points and indices of binding interface points, such that the total cost matrix over the aligned elements is going to be minimized. And this is going to be a loss function that's going to be minimized during model training. Some questions from Joshua. I did not understand how we know the alignment that should exist between Y1 and Y2. Yes, so it's a very good question. So it's because we train them. So our model is based on multi-head attention layers. So it's essentially going to predict a list of key points, right? So we can assume that the first point in the list of Y2 corresponds to the first point in the list of Y1, and that's just because they share parameters in the model. So maybe they find complementary or similar patterns. So they are already matched because of the model parameters.
Okay, I get it. I also had another question. Yeah. Earlier, you said that the message passing neural net would preserve this independent rotation, property. I didn't understand how that is achieved, how you guarantee that that equivariance.
Yeah, that's a good point. I didn't show the actual equations, but what happens is that we update both coordinates and features, and the way we of nodes, and the way we update coordinates is in a way. If you know the paper EN equivariant graph neural networks from ICML last summer, we actually generalize what they do. So we make sure that when we update coordinates, we only have scalar. So it's basically based on forces, right? So we take differences between coordinates of neighboring nodes, and these are going to be multiplied by scalars, which are invariant SE3 invariant scalars, and then we add everything up. So it's really.
I see. So you're saying the update of the numbers that are being associated to each node in the graph guarantees this by construction.
Yes, exactly. Yes, and for features, it's only using invariant messages. So for instance, just distances of 3D coordinates. And for coordinates, it's going to use forces Yeah, so essentially distance between 3D, difference between 3D coordinates. So different vector difference between 3D coordinates multiplied by scalars which are invariant.
Okay.
So it's really generalizing the EN equivariant GNN paper approach, but that was done for a single object. And now we do it for multi-objects and make sure that we have this independent multi-body equivariance. Okay. Any other question? Have you used sidechain information? Only somewhat implicitly in maybe two ways. One is we have these local frame orientation features. So that captures some aspect of sidechain structure. And also even more implicitly based on a residue identity. But yes, this is something that we have to integrate more. What we have done is we have used all atoms, for instance, instead of just residue alpha carbons in our graphs. The problem is that we see massive increase in computation and also in GPU memory. It was training much slower, and we could not get improvements with that. And this is both happening for drug-like molecules, as I'm going to show a bit later, because also this approach also works for drug to protein targets, and it also happens for protein-protein interactions. So for both cases, we could not get we could not get improvements with all atoms instead of just residues. So this is something that we are actively looking into. Just something to mention regarding sidechain information. In PDB-bind and DIPS and other data sets, you do have sidechain structure. But my current understanding is that many of these sidechain structures are somewhat so the experimental measurements might be somewhat inaccurate, or the variance might be very large. So if you base yourself too much on the sidechain structure, you might have quite a bit of noise in your data that you don't want to have. So essentially the backbone 3D structure is more stable, as far as I understand from the discussions with various partners and collaborators. So, but feel free to contradict if someone knows something else regarding that.
So just curious, like follow up on that, like so can you just like use the sidechains? I mean the sidechains like around the binding pocket. So in that case, maybe the computational complexity is still like handleable, right? So there are not many molecules, too many like residues in the binding pockets, right? So if you only use those sidechains, maybe it should still be like manageable, right?
But unfortunately, so in our case, we deal with this blind docking problem. So we don't take any assumption on where the two proteins are more likely to interact. But if you do some other way, let's say, maybe you have a different deep learning model that predicts based on the sequences where the contact area is likely to be, you can then take our model maybe and use sidechain just for those residues which are more likely to interact. So here, what we haven't done any sort of pre-training with sequence pair models that predict the contact map. There are a lot of papers in that direction, and that's a future direction. Yeah.
Right. Yeah, because I feel like if you don't use sidechain, you cannot determine the exact position of the ligand, right? Because I mean, eventually the interaction happens. I mean, physically, the interaction happens between the sidechains and the ligands, right? So I think in the future, maybe that's a really important direction to go in, right?
Yeah, definitely, for sure. Yeah, as I said, we have tried various things so far. I'll show later for small ligands how it works.
Okay.
But yeah, so far we were facing some computational challenges, and also we could not get improvements. But yeah, clearly. But as I said, yeah, sidechains might be, yeah, the data might be somewhat noisy. But yeah,
right.
I totally agree. Okay. One more question. The number of key points are hyperparameter. How many key points did you use? So we tried various numbers. It also depends, of course. We looked at the data and we see, okay, we range between I don't know, maybe 20 to a few hundred, maybe at most 200, something like that. And then we tried obviously hyperparameters in this range, right? So we tried 25, 50, 100. So in the end, 50 was working the best. How expensive is it to train these models? it takes around 4 days on a GPU with 40 gigabytes of memory. So that's one downside for protein docking. You need like 40 gigabytes of memory, even just using the backbone, right? So no side chains. So that's what I'm saying. When we used all atoms, it was like really much harder to. Oh, this is, by the way, batch size 10. So if you use all atoms, you have to use base size 1 because you cannot do more, even on a 40 GPU. So it's really. Yeah, for protein-protein interaction, have you compared with AlphaFold multimer? So AlphaFold multimer was actually released 1 day after we submitted to ICLR. We haven't so far done a comparison. They are doing the problem of really they start with 2 sequences and they predict the final complex.
Right.
So I would say it's a bit different because many times you know the structures, especially in pharmaceutical companies. They know, okay, I have this cancer protein. I have looked at this structure for many years, maybe. So many times people know the structures. I totally believe, and we are actually trying now to build on top of AlphaFold to take AlphaFold predictions and the input to our model instead of just true protein structures. There's a lot of synergies there. I have to say that AlphaFold multimer also uses MSA information, which we don't use here. So maybe that's one advantage of our model. And as I said, yeah, we assume already we have the structure. So we don't want to add an additional complexity in our model to say, yeah, please also predict the 3D structure of the proteins, which is already like maybe a very, very complicated problem in itself, as you know.
Right.
So I guess there are many complementary and also kind of common views of AlphaFold multimer and our model. One aspect is that AlphaFold multimer is actually currently is taking the sequences and assuming a pre-order. So it's assuming I have to order the sequences in some way, first one, second one. And in fact, in our model, we don't have this assumption. So the order of 2 proteins doesn't matter, which is what should be.
Right.
Yeah. So there is just 2 sets of key points per protein pairs. Can they interact at multiple locations? Yes, sorry, if I understand. So what are you asking if the 2 proteins can have different ways to bind? Yeah, that is possible. And that is possible in the data we see. It's one way, right? Usually, I think that's relatively rare, but it's indeed a downside of our current model that we don't predict, let's say, a set of possible 3D structures. So a question there is how much data could be used for this problem. Joshua, you have one more question. Are you assuming that the 2 proteins are only going to move rigidly when binding? Currently, yes. I'll show later that for drugs, more drug-like molecules, we assume we are going to model flexibility in the drug conformation, not for proteins. So proteins are currently rigid. That's something, an aspect that we are currently actively working on. And yeah, definitely, that's one downside at the moment, that we don't have flexibility in the protein. So yeah, we don't have flexibility in the protein structure, but the fact that we don't use sidechain can also be seen as an advantage because sidechains are usually the most flexible parts. So we are using only backbone, and it seems to work relatively well, as I'll show later. Yeah.
Octavian, I just want to make a time check here. Do you think you have enough time to go over the remaining of the presentation?
Yeah. I will.
A lot of questions come in, so.
Yes. How do you believe like 15 minutes maybe? I don't know if people have other commitments and have to go.
It's up to you. If you have a few more time to finish the presentation, it's fine. Anyway, there's recording for people who will have missed the end of it.
Yeah.
Okay, cool.
Okay. So so let me. Oh, there are a lot of questions. Maybe okay. Would you want that I finish at least and show some experiments, and then I'll take more questions at the end? I see many people are very interested here. Okay, yeah. Sorry. I will remember that I remained at Constantine. Oh no, sorry. Gian. Sorry. I hope I pronounced your name properly. Sorry if I messed it up. Okay, so I will get back to your questions later. Really sorry for that. yeah, I'm really excited to see that you are very interested. And okay, so let's. Okay, so this was the pipeline. So we now superimpose y1 and y2, and we recover the transformation. And the way we superimpose it, actually, it's also differentiable. So we run this captcha algorithm. So this is a classic algorithm to superimpose 2 point sets in using just rotation and translation in 3D. So it boils down to having an SVD matrix. So you have to compute a cross covariance matrix of y1 and y2. This is going to then be applied an SVD on top of it. So we use PyTorch SVD, which is differentiable. So we can all backpropagate through this arrow that you see here. So finally, we have a loss function. So we have this optimal transport loss function that I explained before. We have also a mean squared error for the ligand RMSD, the loss function, and we finally have an auxiliary non-intersection loss function. So we sometimes see that we have a little bit of intersections. So let me get into that. So what happens is that we want to give a large penalty to point clouds that intersect, and a low penalty to point clouds that are tangent or are non-intersecting. And the way we do it is that we build this very efficient loss function, which is shown here to prevent non-intersections. So it essentially means, just intuitively, there is quite a bit of math here, but just intuitively, around each residue we build, let's say, a Gaussian. So you can view it as a sphere, right, with some certain radius. And then the surface of a point cloud is going to be given. So we have this mixture of Gaussians, right, essentially. So the surface of a point cloud is going to be a set of points x in 3D for which the Gaussian has a certain gamma value, right? So the interior of the protein is going to be the set of points for which the Gaussian, this mixture of Gaussians, this function g has a value less than gamma, and the exterior is going to be, the outside of the point cloud is going to be the set of g of x bigger than gamma. So now you can imagine that we can simply do a max margin loss function by really trying all pairs of points between x1 and x2. So we want, if we have a point which is inside the other surface, we just do a max margin to make it go away, like get out of, essentially, intuitively speaking, get out of the other point cloud. So this doesn't require any pre-computation, surface pre-computation, which can be expensive for various applications. Okay, so I have finished with explaining how we do the model for protein-protein docking, but I want to argue that this is a very generic model. And in fact, we have already applied it for small drug-like ligands binding to proteins, to rigid protein receptor structure. So what happens here is that we start with a ligand graph. We have an initial random RD kit conformer, and we're going to model flexibility of it. So we're going to model 3 aspects. We are going to model how the drug, where the drug binds. So the binding location, it's again the blind docking problem. We're going to model how exactly, so the orientation and the location, and also the change in flexibility. So we're going to model also the change in torsion angles of rotatable bonds, because we know that local structures are typically rigid for small ligands. So the pipeline is very similar to what I showed before with some additions. So this is what I showed before, but now we replaced one of the proteins with a small drug-like ligand. And instead of taking alpha carbons, we now take coordinates of atoms, individual atoms, including hydrogens, and then we build a k nearest neighbor graph with atoms. Again, a random RD kit conformer. And this actually introduces some stochasticity in our model, because we can change what is the input RD kit conformer. But you will see that there is not a lot of variance when we do that. Okay, so we run our pipeline, we predict some transformed coordinates and features. We do, again, the multi-head attention, we predict key points. This is going to give the rigid transformation. What I don't explain here is how we predict the conformational change of the drug ligand. And that's actually shown here. So the key idea here is that we take the transformed coordinates and we say, we actually want these transformed coordinates to represent the atom coordinates of the final bound conformation. Okay, so we do that by changing only torsion angles of rotatable bonds to fit this Z point cloud. So we have this Z point cloud, which represents now coordinates of the bound ligand, so of the deformed ligand. We train it as such using captcha RMSD. And then as a post-processing step, we take the initial ligand And we are going to just change rotatable bonds in a way that's very efficient using von Mises log likelihood function, so loss function that can be minimized in closed form. And we're going to fit the ligand in this point cloud. So I didn't explain too much on the details of that, but if you're really interested in the mathematical details and how we do that super efficiently, like really in a fraction of seconds, we can do this fitting of rotatable bonds. You can check our paper. It's called EquiBind. Okay, so now let me finally dive into some experiments. So let me first show experiments on flexible drug ligand to rigid protein docking. Again, this is like a blind docking, so it have no assumption of where is the binding site, which is actually very important for many downstream problems. As data set to use PDB-bind, which contains roughly 19K complexes of proteins and drug-like molecules. We found it very important to have a test set which has very recent release structure. So 2020 version of PDB-bind, and trace set is everything before that, before 2019. And moreover, we also removed shared ligands between train and test. So yeah, this is also to prevent overfitting. We do have some common proteins, but not in the visualizing example that I'll show in a second. As baselines, we have really some of the most used academic open source baselines like Mina and Autodoc Vina. On top of those, so this rely on this idea of heavy candidate sampling and scoring. And there is this deep learning based model which has been using, it's called Nina. It's using deep learning for the scoring part of these pipelines. And there is also this Glide software developed by Schrödinger, which is a commercial software, and again, very used by for instance, many of our pharmaceutical company partners at MIT. So I'll show just some experiments here. We have more in our paper, so feel free to check them out. So here are the baselines. First of all, so we have Q-Vina wide. Wide again means blind docking, so no assumption on the binding site. We have Nina, Smina, and Glide. And we have ligand RMSD and different percentiles, and also the percentage of molecules predicted as being less than 5 angstrom to the true to the true structure, and the 2 angstrom from the true structure. So you see here the plots. Obviously, we want the plot to be as high as possible. Okay, so these are the CDF plots, right? So now we have EquiBind our model. You see it's much faster than the existing methods. And it often outperforms the other models, or it's on par. With one exception, if you look at the 2 angstrom here, we are actually behind. So this is really like the fine-tuning regime of this problem, of this task. So what we can do to also solve that fine-tuning regime is to actually take our predictions and now fine-tune them with. So Q is like Q-Vina wide, is this model, and S is Mina. So Q-Vina can be run in different local search strategies. It's called exhaustiveness. So how much you want to let it search, right? So of course, if you want to let it search for more, it's going to increase the runtime. So it's always a trade-off between runtime and quality. So you see that if you let it run more, it's going to improve. And finally, if you take Smina, you are going to get an increase in computational cost, but you are going to get the best numbers overall. So you're going to improve in really all numbers. So some qualitative visualization. I just selected one very nice visualization. So when we had this project more or less done, we took it to one of our partners from Relay Therapeutics, and they were actually very excited to try it on one interesting cancer protein. Is called 6HD6 in PDB. So it's actually tyrosine kinase. It's responsible for 3 types of cancer: lung, leukemia, and gastrointestinal tumors. And only three drugs are known to currently inhibit this protein, if you look in PDB-bind. And 2 of them were developed by Novartis, and one of them was actually ABL001, was just approved like very recently, end of last year. So this protein was actually not part of our trains. So we removed on purpose this protein from our train set. And also the ligands, of course, as I said before. So what you see here, first of all, I'm going to show these 2 drugs developed by Novartis. They are actually their ground truth co-crystal structures are shown here. And if we use Ganea, this deep learning baseline is going to predict both in the same pocket. So that's maybe not so good. If we use Mina, it's going to predict again both in the same pocket but the other pocket. If we use Glide, it's going to find the two pockets, but just for some reason swap the two ligands. If we use our model, it's actually going to give our model fine-tuned with Mina is going to give very good predictions and is going to find the pockets very accurately. So this is what one example that our partners were very excited about. Okay, so let me conclude by showing some experiments also rigid protein docking. So here we use two data sets. We use DIPS, which is a recent data set from PDB. It contains like roughly around 42K proteins. And there is a classic DB, it's called docking benchmark data set, which is now version 5.5. This is a very, very small data set. One thing to keep in mind is that DIPS actually only contains the complex structures. It doesn't contain the unbound structures. And that's one of the reasons why we tackled the rigid protein docking in the first place. The DB5.1 contains mostly rigid proteins, contains some flexible proteins, but a small fraction of those. So we use some baselines, again, academic and commercial baselines. ClassPro, for instance, was also used in AlphaFold multimer paper. So in terms of speed, so you see here these baselines. What you have to keep in mind is that the numbers of runtime for ClassPro and PatchDoc, they are pessimistic in the sense that we had to submit test sets one by one, test examples one by one on their websites. Because there is no way to run it locally, or we couldn't make it work locally, and the others that were run locally. So our model is much faster because it doesn't have this heavy candidate sampling strategy. In terms of quality, we compare with two metrics like complex RMSD and interface RMSD, and you see here that we are better than 3 out of 4 baselines, somewhat better, with the exception of HDoc. With HDoc, something is maybe a little bit like it doesn't really favor a direct comparison, I would argue. I will say why. So HDoc is a template-based model technique. So it uses templates from known docked structures. And the problem is that the way we sample our train test, let's say for DIPS, is really based on family proteins, but it's not based on release time. And in this way, it's really possible that some of these baselines have used templates from our test set, let's say. And in fact, when we looked at the distribution, you see here like HDoc seems quite bimodal distribution. In fact, if we look at the numbers, I don't have the histogram here, but really exactly half of our test structures, like 50 out of 100, are 0 point something RMSD, so almost perfect. And the other half are really like 10 or more angstrom. So this kind of maybe hints the fact that this template-based modeling might not be favoring like direct comparison with machine learning models. Anyway, so just want to finish with some visualization. We have a ground truth here of a protein complex. We're going to keep this black protein fixed across all these images, and you see like this model, these baselines, they place this other protein in the wrong side of the black protein. Our model finds the right side. It's obviously not perfect, as you can see, but it gives a good prediction of the binding site. I did have some slides for generative models of 3D molecular conformations, but I will not have time to go over them. And I'll probably end here to take questions. Sorry, I had a lot of other slides here, and maybe some interesting aspects for future work, which unfortunately I'll probably have to skip. I just want to thank my collaborators and sponsors. And with that, let me just take your questions, and thank you for your patience.
Thank you so much, Octavian. I think if people have questions, they can raise their hand and ask their question themselves instead of writing in the chat.
I can probably go over the remaining questions that I didn't. There are a lot of questions. I remember I stopped after Joshua's last question, so I can go over them, right? This makes sense.
Yes. Please.
So Gian, I hope I pronounced your name properly. Have you thought of integrating equivariant features in the message passing routine using steerable counterpart of the Euclidean GNN? Are there SST equivariant features in the data that could be useful? Yes, sure. There is a lot of work for instance, from people like Taco Cohen and Max Welling. And we do use, as I said, yeah. So yeah. So for instance, and also Michael Bronstein and other folks, for instance, the Massive paper. So dealing with like mesh convolutions, right? So taking the input as a mesh, not just as a spatial graph. So I think these are really complementary strategies. So I really believe the future is really to complement these strategies and create a really strong, powerful technique that can take proteins with their different views, maybe be it like sequence, be it spatial graph, be it mesh of an irregular object, and apply different techniques there. Yeah, so totally, I believe that as a community we can really do very exciting breakthroughs and combining various techniques. So again, I'm not claiming that my model is ultimate, not at all. I really believe in complementarity, and I really believe in collaborations and the way we can together as a community drive drug discovery and accelerate drug discovery, right? So help pharmaceutical companies and academic labs and industry labs to accelerate drug discovery. Have you looked into constraining the binding interface? For antibodies, the binding is usually mediated via a specific set of residues. Yeah, for sure. So this is something we are currently looking at. When you have information about the binding site, which you do have for many classes of proteins, like antibodies, you said, and there are so many others. Yeah, so we are actively looking into that. Pat says, what tells us that the sparse mapping between key points and binding interface points will be the same for each protein pair? Is not the same. That's what optimal transport is doing, and that's why is it nice, because optimal transport is tailored to find always a different matching, different alignment between binding interface points and key points. Since the ordering of points in P sets depends on the order of residues in the protein pair, in my understanding, the ordering of the PIs is random depending on the way residues are ordered in the dataset. It is random, yes, but their bijection is clear, is fixed, right? So that's what it means. I represent it as kind of a list. What I want to mean is the bijection is fixed, right? Sorry if that was maybe unclear from the slides, but they are sets. I said at some point that they are sets. However, in EquiDocs, there is only one mapping that is learned between YIs and PIs. Yeah, as I said, it's one mapping, but it's mapping between sets. So I hope that clarifies your question. What will happen if, Tony asks, what will happen if you input two proteins that don't bind together? Do you predict some kind of binding energy? Yeah, so we don't have, currently, we are working on that aspect, and I don't have anything to share at the moment. But yeah, so our assumption currently is that they do bind, and we train on data that binds. So we are actively trying strategies to incorporate decoys, if you're familiar with the terminology, and so negative examples essentially. Dominic, no, this was a comment for Joshua. Maxim, hi Maxim. Thank you for the talk. Interesting approach. What EquiDoc code? Oh yeah, it's available. I didn't show it. Wait, here. Sorry. Feel free to check our code and try it out and give us feedback, please. And yeah. Oh, someone pointed. Thank you. Falk. In EquiBind, you assume that your protein is fixed and your ligand is flexible. If the binding pocket contains an intrinsically disordered region, the position of the dog ligand depends on a specific position on the disordered region, which is not necessarily the same, like the most occupied conformation of this region in its upper state. How do you deal with disordered region in the protein? So yeah, so currently, as I said, we don't model flexibility in a good way. It's just somewhat implicitly via the fact that we have only some features of side chains. We don't constrain side chains to be rigid. because we use backbone information and some sidechain features. But we're working on that. Maybe using energy-based models. That's one way, and there has been some recent work on that. Yun Chao, thank you. Oh, sorry. Maxime is asking, like the fine-grained structures of 2,5 angstrom. Yeah, I showed how we do that and how we fine-tune, and we get very good results. Florian, what is your model doing with? Okay, I already replied to that. Yeah, we are working on that, as I said. Okay, so I guess that's someone else has any other question. Feel free to reach out, by the way, if you have any things, like just send an email, Twitter, you know where you can find me, and my collaborators, obviously.